as long as you have enough detail in here to go into the mechanism, which you do, but you don't have too much detail that you have a whole range of other different molecules and enzymes that don't talk to your poster at all. And that's why it's good that you make your own and don't get one straight from the paper. So that looks good.
Okay.
mGlu5. So what does this mean? So cannabidiol, one of its functions is to produce these endogenous cannabinoids and come in here. Is that what you're...
This is meant to be in here. This inhibits this, apparently. It inhibits FAAH. But
FAAH is one of the enzymes that promotes or catalyses the production of your endogenous cannabinoids, yeah? Is that what this diagram is showing?
no, it's meant to break it hydrolyzes.
Okay, then that
this
makes
into
sense.
that.
Yeah, okay, so that makes sense. And then here, what's going on here?
I don't know. That was
Okay,
more-
so this one's not exactly our part, but
Okay.
this is where she's adding in the mGlu5.
Right.
And she just wants to make sure that this makes sense, and the hardest part is just trying to figure out if this all ties back into the CB1, because right now we know we need the CB1 in there, and we know we have the mGlu5, but we're just trying to really bring it back.
So, okay. Say this is your synapse. You can take this with you.
Okay.
So you've got mGlu5 sitting out here, and you would have glutamate being released up here normally, binding to mGlu5. It does this whole downstream pathway that you've got. But one of the consequences of mGlu5 stimulation is the production of your endogenous cannabinoids.
Okay.
Yeah? And then CB1 receptors are located up here.
Mm-hmm.
So these endogenous cannabinoids then retrograde, travel back to the presynaptic. They bind to the CB1 receptor. It then inhibits further release of glutamate.
That makes sense, because in autism there's an excess of M-glutamate, yeah? This?
I think there's controversy. I
Okay.
think you've got a body of people that think there's an excess, and I think you've got a body of people that think it's dysfunctional.
Okay.
So you obviously only have so much space, and you can't
Right.
go through every single theory out there. So if you kind of want to focus on one angle, that's fine.
Okay.
So have you kind of been working on the fact that there's an excess of mGlu5?
Yeah.
Okay.
That's just what we've found, but it's like you have said, it's hard because there's so many different areas where people say different things and you don't exactly...
Yeah, and I think with autism it's hard because people are researching autism, but they're also researching Fragile X and kind of treating them as one and the same, and they may not necessarily be the same thing.
Because, when I was reading, I also it affects GABR3.
GABA?
Yeah.
It's just if your receptor's made up of lots of different subunits, it's just one of the specific proteins of a GABA receptor.
Okay.
Yeah.
So we don't go that far, because I wasn't sure if we're talking about autism, if it affects these ones, and therefore there's something the stream isn't working, or
so let's stop here for a minute and just go back to the cannabidiol.
Yeah.
What have you got as how that works
So what we have
as
is
its mechanism?
this box.
Yep. Cannabinoid receptors, endogenous cannabinoids that bind to these receptors. Okay. Cannabinoid itself is a negative of CB1s. Yeah, that's fine. I mean, CBD is pretty people are still trying to figure it out. So there's probably 4 or 5 possible different mechanisms.
Yep.
But this is fine.
That's fine.
To
Okay,
narrow in on one. Okay, so what were we talking about before?
cool. CB1,
So it's definitely...
right?
Yeah, so like, because this does GBA, GABA, and then this does something with GABA as well. But I wasn't sure whether they were linked, because in autism they say this is like a chromosomal link. Because when we look at these, all the studies just say we gave them CBD and they got better. And then I'm
That's
like,
handy.
yeah, it's like
Well,
their
it's their
anxiety
social-
improved
yep.
or something.
Okay.
But to me, that's not like
A mechanism.
neuroscience.
Yeah.
It's just,
So yeah, you're right. I mean, I guess I've tried to steer clear of the GABA because I think it probably gets too big and complicated and kind of just sent you down the glutamate path with the mGlu5. But if we draw another synapse here, which is a GABAergic synapse, you've also got CB1 receptors on those GABA as well. So the CB1 receptors not only regulate glutamate release, but also GABA.
So
release.
potentially we could draw that, and then would it be safe to say that with this binding, with the cannabinol there, it would help with the like social anxiety that comes from the excess or decrease in that?
Okay, so if you've got it binding here as a negative allosteric modulator, so it's going to inhibit.
Okay, right. So that would be
Yes.
excess.
So it's going to result in an increase in the amount of glutamate being released.
Okay, so then it would be
Because normally this would inhibit glutamate from
Okay.
being released, but
Yep.
you're blocking that. So you're preventing the inhibition, so you get more release. Does that
Okay.
make sense?
yeah.
So therefore you've got more activity of mGlu5
Okay.
happening. And if you look at the more recent studies of mGlu5 autism, they tend to suggest that the function of mGlu5 is reduced
Okay.
in autism. But there is conflicting stuff. And I think if you're working off the hypothesis that CBD is a CB1 NAM, it makes more sense that it would be effective if mGlu5 is dysfunctional.
Okay.
so it is okay just to use like the Fragile X studies and like the ones like
Yeah, so
all
I guess
the
there's
ones
2
on
studies.
epilepsy.
There's 2 studies that I'm thinking of. One, the author is Chris Pentelis. I think he's like maybe the last author. And it's a really disgustingly complicated genetics paper
Right.
looking at GRIM5, which is the gene for mGlu5. I don't think it's in Fragile X, it's autism. And they said there's a problem with this gene in autism. And then there's another group of people that used brains from people with autism. And they also used mouse model where they knocked out mGlu5 as a model of autism, and kind of suggesting that it is a decrease in function of mGlu5 that's associated with autism. So that's the last name of that person's Chana. So I don't know if you came across any of their papers. So he had a human brain paper
Okay.
for autism.
Autism, yeah.
Yeah, because if this is down-regulated, because this doesn't only just regulate endocannabinoid release, it also causes calcium.
Right.
Influx
And you
and-
added in calcium somewhere, didn't you?
Oh, it's on this chart, but that's because something about calcium does.
Yeah.
Reduces that.
You were
Yeah.
you were wanting to make sure that that was correct though, right?
Yeah, so I think here, this is really good, but I think you also want to indicate that the calcium is not just causing this to happen.
Okay.
The calcium's also causing a lot of other downstream things. So it's causing not excitotoxicity, but like a postsynaptic potential, because it's getting that positive influx here and causing a graded, so stimulating the neuron. But it's also that calcium, and you haven't learnt this yet, it's in a lecture in a couple of weeks with a guest lecturer, the calcium's really important. This mGlu5 mediated calcium is really important for learning and memory, and a lot of those kind of higher order functions. So if you could, I would just have another arrow there indicating some of those downstream.
Okay.
So it's not just
Yeah.
the CB1 kind of thing.
Okay.
Um,
So I guess just our last thing is we left enough room here
mhm.
for a meeting with you and whatever else we're missing. So what would you say we should add? Should we add that as a...
Well, what's this? Because this would... Because, like here, this is essentially this,
Mhm.
except this is just in a very generic manner, not specifying the neurotransmitters,
Right.
whereas this is focused on it being a glutamate
Yeah.
synapse. So I don't think you need to add this, but you could consider modifying that.
Okay.
What's this? That's just telling you what the CB1 receptor
Mm-hmm.
does. This is interesting, how it's involved in... Okay, so how is this important to the story that you're creating here? So,
That's one thing that we were... Because this was created before we had talked with you about the
right.
mGlu5.
And you don't have to have the mGlu5, but it just gives you
It definitely
an avenue
helps.
to
Yeah.
go down. Yeah.
So do you think that that's still necessary?
Without really taking the time to
Yeah, break
read
it
it
down.
in context, in total, it just looks like it's thrown in there.
Okay.
Like it, to me, this, I'm not sure how it fits in
Mm-hmm.
with
Okay.
the story that you're, especially if you're then kind of saying, well, CBD kind of feeds back, binds to the CB1 receptor. This affects what's going on here with the neurotransmitter release. I'm not sure how that then relates to this.
Yeah, I think that at that point we were going to focus on CB1 and
Okay.
CB2,
Yeah.
but it seems now that we're not really going that pathway as much.
Yeah. So, I mean, you can still have it. Now that I look at it in more detail, I can see that you've got your voltage-gated calcium channel here,
Mhm.
and that's kind of what's happening here. So the CB1 receptor here inhibits the voltage-gated sodium channel, which is what this is showing, and because you have decreased calcium coming in, that inhibits this from happening. So this part of the diagram is
Is-
what's happening
yeah.
here. You just don't have the voltage-gated
Right.
channel drawn on there. This part, yeah, I'm just not sure if it's,
Necessary. Okay.
yeah, like in the bigger scheme of figuring out autism and the pathology and what's going on, I'm sure this is very important, has relevance, but in the context of what you're presenting here, I'm not sold.
Okay.
Yeah.
Sounds good.
So is there any evidence that you've come across that CB1 or the endogenous cannabinoids are altered in autism?
I found one thing that they measured the plasma concentrations
Okay.
of AEA and
Yeah.
they found them to be lower,
Okay.
but I don't that's where that study ended. It was
Right.
just a sampling. And
Okay.
then a lot of them, because they use then THC,
Yeah.
so
Yes.
some of them
Yeah.
aren't like relevant, or they'll
Okay.
say that the THC
Yeah.
like doesn't like it does something else
Right.
with
Yeah.
both.
Because I guess you're kind of saying, yay, cannabidiol is great treatment for autism, and there's so many different avenues you could take it down. You could say it's a good treatment because the brain's own cannabinoid system in autism is dysfunctional, or cannabidiol's another important thing that it does is it helps apparently reduce brain inflammation. So that could have been an avenue that you went down to say, well, hey, in autism there's all this evidence that there's inflammation. This is how cannabidiol regulates
Mhm.
inflammation. The mGlu5 side of things, yeah, if you can kind of go down the path and say, well, hey, there's evidence that mGlu5 is altered in autism, and CBD works on this pathway, and it might be rather than fixing a problem with the cannabinoid system per se,
Right.
CBD might be helpful as kind of a target to get to the problematic mGlu5. But I don't know if there's any evidence on CB1 or part of the cannabinoid system. You might like to consider adding that in.
Okay.
That might.
So that could fill in our crack there, and we would still probably want to leave that in there if we focused on, or
Yeah.
still no.
Well, I guess you explain to me what it's. Yes.
Yeah.
If you can.
Make it more relevant.
Yeah. So, if there's any evidence that in autism there are problems with neuronal number,
Okay.
and you think it's driven by problems in cannabinoid system signaling through this pathway, then it's relevant. I think I'm pretty sure in autism there's evidence, I don't know about neuronal number, but synapse number tends to be higher
Okay.
rather than lower. So I don't know if neuronal number's also higher, because it's thought it, because when our brains develop, we have all these, what are they called? Neurons.
Mhm.
All of these neurons and all of these synapses kind of overdeveloped, and then we kind of prune back and refine, and it's thought that process is a little bit altered or not
Okay.
working. So it's thought they're left up with too many connections that it's very hard for them to filter what's appropriate and what's not. But yeah, if you're going to have it in there, you have to explain its relevance.
That makes sense.
Yeah.
Okay.
So what's going on over here?
So that is just her explaining how the receptor changes shape when CBD is binding.
But this is the one that Rhiannon didn't like it. As well, anyway.
Okay. So it's kind of,
So it's saying it's a negative allosteric modulator.
yeah.
It changes the structure of CB1, so other cannabinoids can no longer bind to
Right.
CB1.
Okay. That's fine. It's a lot of space devoted to.
Yeah, but I
Something
think that
that-
if we take this out,
Yeah.
cut this down,
Yeah.
we'll have this space still
Yeah.
to focus, and then the conclusion to focus
Yeah.
on.
Yeah. And you might like to. Because you're gonna have a little bit of space.
Yeah, I'm not sure how we should fill that space at this point where we.
Yeah, you may need to add an additional mechanism.
Yep.
Otherwise, it might be. I think it's too difficult to go any more in depth of this one.
One option is listing the
Limitation.
yeah,
Mm.
or is that not worth it?
I think with the amount of space that you're going to have, that might be a little bit superficial. Like this will pass, no problem
Yep.
with it passing.
Well, but we
Um,
want a good grade.
Yeah, okay.
Not just
then
a pass.
Then I think you probably want basically another one of these.
Okay, another
um,
flow chart.
Whether it's related to the GABA story
Yep.
or inflammation, or what's this? So is this the general process, or is there evidence that any steps of this process are disrupted in autism?
Well, I was just focusing on the angle that this was meant to be decreased, the AEA.
Right, okay.
But
Based on that serum study?
yeah, plasma study.
Yeah, okay. But you don't know if anyone's looked at this in the brain tissue,
No.
the CB1?
Because a lot of the studies just say, like, we can't get brains, so we're not.
Autism brains are really hard to get.
And, like, there are, like, mice knockout models, but then a lot of them do like the syndrome more than like autism.
Yeah, I would definitely include animal work in here
Okay.
because it's really hard to get at a mechanism without including that in there, and there's obvious limitations anytime you do anything with animals. But I guess the trade-off with that is able to get access to the tissue, the brain tissue, and figure out exactly, in this particular genetic mutation, for example, this is what's going on. I don't think for the mGlu5 knockouts, I don't know if they've looked at the cannabinoid system, but that would be nice if you could create that link to say, well, at the genetic level, there's this identification that GRIM5 is altered, and GRIM5 is so important for cannabinoid signalling that is altered, and studies have actually shown when you disrupt mGlu5, cannabinoid systems all over the place. So it's not a direct autism study, but it's a mechanism study. But I think I would maybe look at either GABA or inflammation. I think you want another mechanism. Yeah. That's probably not ideal
Sad.
news on Wednesday afternoon, to be honest. But I think that's where you're at.
Okay.
It looks good though. Apart